way out on a tail from what the other things have been. I don't know anything about making money. These are all just questions of answering biology questions. But I teach freshman biology. I'm used to a skeptical audience, so I will entertain you as we go here. These are freshwater mussels that I work on. I'll get into the sort of the analytics side that we deal with. I probably won't be as impressive as you're used to. I'm coming at this more from the biology side. All of those that I showed up there were ones we find here in Wisconsin. There's about 60 species here. I want to be talking about global patterns and Africa because that's where I like to work. So I'm going to start out today just giving you a little background about these animals because to me that's the important part. Talk about the kind of data that we've used for forever, since before there were books until about the year 2000. That's really kind of when things changed. So how the kinds of data that we have and where they are. Talk about some resources that have become available recently, and then our solution to this problem we call the MUSSEL Project Database, which is sort of for this project that we have going on. And I just wanted to define some terms that how I use them, because maybe they're different than you use them. By big data, I just mean data sets that are too big to put on index cards and deal with sort of manually. That's what we're talking about. And I also want to make sure we talk about this word here, bioinformatics. There's the Wikipedia definition. Bioinformatics, as a general sense, kind of covers what I'm talking about, but really if you say that to a bioinformatician, they're talking about DNA, genomic data, biomedical applications. I'm not doing that. And so I usually like to refer to this as biodiversity informatics is what we're talking about. I'm sort of limiting it that way. I'm going to say we a lot. When I say we, I'm talking about my collaborator here, Kevin Cummings at the Illinois Natural History Survey. We've been working on this project for a couple decades now. Over the internet on this project as a digital project, but also here we are in the Congo River working in the field. So let's just get into it here. Freshwater mussels are one particular group of bivalves. You might call them clams, although that's not a good name for them. There are specific kinds of bivalves that are clams that live in a marine environment. These are all freshwater. If you know everything you know about biology from SpongeBob, then you probably envision these things to be kind of like a person, only just with a different shape, but with eyes and a mouth and everything the same. They're not. Okay? They have two shells that have a hinge on them, so they open and close. So we're looking at this thing from the side. This is its left side, its right side is over there. I'm just going to take its shell off there. And you can see that this is a very different looking animal. There is no head. It's basically a living rock. It lives with about this much of its body embedded in the bottom of a river. They're sedentary. They're built very much for comfort and not for speed. They have some muscles here that they can open and close these two shells or valves. That's where that bivalve name comes from. They have these very large gills right here that they use to just sort of pump water through their body, and they're filter feeders. So they're just capturing little food particles from the water and passing them to their big lips here, their labial palps. The mouth is right there. There's really no head. It really just sort of an esophagus just ends in an opening. They have a little foot for burrowing. So it's a very different kind of animal. Not much in your vertebrate experience would help you relate to what they have going on. They're interesting because besides being different from us, they have a parasitic life cycle as larval stage. So even here in the Wisconsin River, we have separate sexes. That may not impress you at all as a mammal, but lots of animals don't, but these do, the male and female. The male releases the sperm, fertilizes the female's eggs. The little tiny larvae will actually attach to the gills of fish. They'll make a little cocoon there, they'll go through their metamorphosis, and then they'll come out and grow up into a big adult. So it's a very interesting life cycle, something we've studied a lot. If you know anything about freshwater mussels, you've heard about them in the news because they are endangered like most freshwater animals are. For a long time, just here in the United States, we've known, these are old references from Coker here, as soon as we started impounding rivers, we start changing the way water flows, we move their fish around because of that, we affect the mussels. Also, until about World War I, the little white buttons that were on button-down shirts, that's what they were made of. That barge you see right there is full of mussels that were collected. These things live for decades, so it's a very slow life cycle. So we've hunted them to extinction almost. It's also the case just worldwide that people live by fresh water and we do a lot of damage to it. This is the port of Brazzaville on the Congo River. It's just garbage everywhere. This is the Rio Ataya in Peru. You can see people living and using the water right there. For lack of a better word, that's a washroom. We have impacting freshwaters, and this impacts freshwater animals. So this is sort of what drives my research. This is what drives funding for this research, is we need to know about these animals to be able to protect them. Okay? So in trying to relate the kinds of questions that I answer to people who don't answer those kind of questions, I'll tell you, my subdiscipline, what I'd be called, is a systematist. I am interested in how organisms are related to each other evolutionarily, how they're distributed around the planet, what the system of classification is for those things. That's where that name comes from. Try to think about the kinds of questions that I want to answer with the data I'm going to talk about. I think maybe the easiest way to relate is to think about like a field guide. You've seen those, maybe not for freshwater mussels. This is just one we made to give away to people in Africa who were helping us. But the kinds of things that you'd look for in a field guide and how it is that we would gather the data for those things. And those things, I think, are what you'd expect. We'd want to know how to recognize this animal, how to recognize it from other animals like it, where it's found. So here's a map. This is sort of, we call this Zambongo because this is the Zambezi River and this is the Congo River in Central Africa. So it's covering this region. That's the name of that book. So where it's found, some information about where to find this thing, and what to call it. Those may seem like trivial questions, although if you just pause for a second and imagine that you are Adam and all the animals are in charge of you to name and describe where they are, Texas is about this big right here. So how do you do this for all the animals that are there? Okay? Now I'm going to deal with each of these three questions, especially these two, what to call it and where it's found. You'd think that this question might be really easy, what to call it. But actually, people working all over the world talking about the same things. We actually have this really strict set of rules that requires us to know what an animal has ever been called ever by anybody since words have been printed. This is the International Code of Zoological Nomenclature. So right there we have a big data problem. To name an animal, we need to know what it's ever been called. We can say the rule is simply the oldest name that's the correct one. But that often takes quite a lot of work, and we often discover older names, and we have to change the names for things. So I'll talk about that. This has been a big data problem for us. And also the question of where it's found. And this is going to lead to the important idea of collections. We could imagine that we could go right there and do some sampling there and find the species that live there. And that's what we obviously need to do that. What do they look like? What do they do? What's there? But it would take us forever to cover Texas in there. So how do we do this work? We can go in the field, but actually more of our data is available in natural history collections. If you've been to the Smithsonian, you've been to the Natural History Museum in London or out in LA or in Paris or in the Carnegie in Pittsburgh, you've probably seen the exhibits and the dinosaurs and things like that. But the bread and butter of what these museums exist for is actually to house specimens as evidence of species being in a certain place at a certain time. And so we call those vouchers, and so an example here of data that we've always used would be specimens collected. These ones here, they look like this. This has a name on it of what the people called it in the field. Here's some original labels and handwriting and things like that. A locality, Zambie in the Belgian Congo. These are our data. These are our data. A specimen with locality information. We use these things to know where they're found. We use these things to study their anatomy and compare them. We would do this for any group of organisms. In a mammal collection, there'd be a bunch of mammal skins. In bird collections, a bunch of stuffed bird with just cotton balls for eyes. This is how we do this work because I can go much more easy to Europe or Washington and cover a lot of ground. And you'll see how much data we can get by investigating these things. Museums also have libraries. If we have to know the names of all these things that have ever been called to figure out what they're supposed to be called, then we need libraries dedicated to that purpose. And until about the year 2000, that's when I would say the transition happened. I got my PhD in '97, so I was working sort of across this transition. What we did for a living is that we traveled from Washington to Cambridge, to Philadelphia, to London, to France, just sort of cycling through, going through these thousands and thousands of specimens over the course of our career and using these libraries to get that work. That's what we had to do to get these data, because it really is a lot easier to do that than to travel to the tropics or wherever we're working to just do our little bit of sampling. And I'll just point out before I get into our specific project here, with the internet, a lot of data have become available to a lot of people. Okay? Number one, every museum, every major museum has all of their whole catalog that they have is basically served through the Global Biodiversity Information Facility, what we call GBIF. But it's just lines of text with that information that was on those labels. You don't know what it was. You don't know who put that name on there. You don't know what data monkey made all kinds of data entry errors. When we were doing this in the early 2000s, when I was in Philadelphia, it was just people paid $7.50 an hour to sit and type in giant ledger catalogs. But we do have a lot of information available about different kinds of organisms in different places. Mapping has become so much easier that now we can, if we can get Latin long, although generally our localities are descriptions. The best locality that we could get that's for a human is the Wisconsin River, Stevens Point at Buchwald Park in Portage County, Wisconsin. That's really handy, but that's not that useful for a computer. Nowadays, even Wikipedia is among the best sources to find the Latin long of localities. That's changed just in the last 10 years or so. Google Mapping is very helpful. Almost every natural history biodiversity publication published before about 1930 is available on the Biodiversity Heritage Library. So we've gone through all of those big libraries so that those descriptions, even back in the days when everything was in Latin, we can find that information. It's all available. There have been multiple initiatives like the World Register of Marine Species, WORMS, or the Integrated Taxonomic Information System. That's our government's plan here for getting the names of organisms, how they're classed, just lists of names of organisms of diversity there. This is the International Union for the Conservation of Nature Red List. This is endangered species worldwide. So there are all of these new sources of information that we can pull together, including information about DNA. So anytime anybody, biomedical, biodiversity, does anything with a DNA sequence in a paper, it's all uploaded to NCBI so that the genes of whatever gene for whatever taxon is going to be available there. Those data are there, and the National Science Foundation gives us some what we call the Terra Grid that we can use for fast data analyses. All of this has transformed how we do our work quite a bit, although probably not as much as you'd expect, and I'll talk about that in just a second. One of the things you find is that if a lot of people came here talking about biodiversity research, this is the part they would talk about. This is just by year, starting in 1980s and going here. The rate of publications dealing with DNA sequences, using those things to discover relationships among animals and plants, increases exponentially every year. That's where most of the work is dedicated, because actually, although that sounds really technical, way more than reading books and going out looking at shells, this is actually super easy. DNA is very amenable to using Python or Perl in the olden days because DNA is words, it's text, and so we can work with it very easily, and the same tools that we invented to have our servers give us messages now and then are the same ones that we can analyze DNA with. Okay? So, what can we do with all this data in a biodiversity context, or what good is it? There are lots of benefits that are obvious here, I think. Number one, by making all of this data available, it's much more democratic. This used to be limited to people who lived in major cities with major museums in Europe and North America. So we call this repatriation of data. That people now in Africa, in Southeast Asia, in South America have access to all of the colonial collecting that happened in the 19th century and can work with those specimens. So lots more people can work on these kinds of questions, and we'll see that in the rise of publications that have happened. There's so much less duplication of effort because so much is being put online, so that if I want to do a study on the genetic relationships of the mussels here in the Wisconsin River. Most of the work has already been done just by someone else who's put the sequences there and made it available, or has made their collection data available so that I don't have to travel there. One of the things that I like to play with is that when you build these comprehensive databases and you get them together, right, for just pulling the data together to answer scientific questions, they have emergent properties. You can start answering questions that you didn't even know to think to ask before you had the data together. And so that's the kind of things we'll play with. And I'll show you some of the analyses that I've done. What I'm more interested in is what I learned from Homer were called crisotunities. Chinese apparently have the same word for crisis and opportunity, crisotunities. These are the things that maybe the problems that we need to solve that we want to work on. Number one, electronic versions of data are not the same as the real objects. And oftentimes a photograph isn't enough, or just having a text piece of information isn't the same. I know a lot from the handwriting of the person who wrote it and those sorts of things. So often having the objects matters quite a bit. As with everything else, the data are being generated way faster than anybody can get to it. So we can dump all of every publication since 1758, when things started really counting, on the Biodiversity Heritage Library, and probably most of it nobody ever looks at because it really is just naked data. But what I find when I'm, as a biologist and dealing with other biologists, is that if you don't have analytic skills, then you can only use the tools that someone else has made for you to do this work. And so what I want to talk about are the tools that we've developed to do this work ourselves. We call this the MUSSEL Project Database. We've developed it for the data entry purposes. We use FileMaker just because it is a breeze for automating data entry and doing that sort of work as an interface. Because basically we have built these data, not pulled them offline. We've typed in from books. We've worked in collections, but then we use Python for most of the analytics purposes, so we just export that and work with that. National Science Foundation wants me to tell you that they paid for this. All of our data in our ERD here, we're compliant with what we call the Darwin Core. For decades, we've had a data standard for dealing with biodiversity heritage data, so databases can talk to each other, and all our data are compliant with that. You can go to and it's all very, very intuitive. It hardly seems like we need a data standard. I'm not going to talk about my ERD here, but you can go to our website at themusselproject.net and find out if you want to learn more about it. I just want to talk about some specific pieces of data and how we use them and how we've changed how we think about things. So like I said, with any particular freshwater mussel or any animal, any plant, they've been named multiple times. We call those described species as opposed to valid species. So in our database here of described species, I have a name here, Coelochura mies africa. This has been described by Pillsbury and Beckar. We have the publication where it's fine. That little E means it'll take it right to the publication so you can see the description, see their figure that's in there. We also recognize that that thing is actually just another name for Coelochura gabonensis, that this is the valid species. This is a real thing in nature as far as we're concerned. This is just what we call a synonym. A name that has just been an extra name. The thing is, with freshwater mussels, it's about a 5-to-1 average. There's about, on average, 5 names for every one thing. There's about 400 species that are like only known from their descriptions. They've only been named one time. That's the only time they've ever been seen. There are 2 species in Europe, just 2, that account for about 550 names. So there's a lot of variation in there, and you can see that that's a data problem to sort that out. These are what our data look like. So this is just one pair of facing pages here from Daget's book where he's saying this is a species, and here are all the names of other things that fit under this. We call this a synonymy. All of these are synonyms. They're different words for the same thing. Okay? So those are our data. What we have that other people don't have is that data, like I showed you, we've gone through those publications and actually not just had our opinion about how this name fits with the real world, but we record everybody else's opinions as well. So we have the history of how this name was treated. And I just sort of picked a spot here where you can see that Haas here treated this thing as valid under some weird name, mies africa, mies africana, but everybody else has considered it a synonym there. So our database right now of freshwater mussels, there are just under 5,000 names, for just over 900 species. This is a data names move from one species to another. Different people have different opinions. That's why we have that history. We have about 64,000 opinions from 1,000 publications here that we can talk about those things. And having this allows us to actually look at, well, let me just say, all this is on our website as well, so that all of that information is available to everybody. If you can use Excel and you can get a list, people have been doing this forever. I've been seeing these plots since the 90s. For example, what I'm showing here is just an accumulation of all the names over time, just a stacked accumulation. So we're filling up here to almost 5,000 names, starting with the first one being named in 1758 right there by Linnaeus. And we do the same thing, which is just, it's so boring. There's all the valid names. So these are the names that are really in use and when they accumulated over time, which is just so arbitrary. It's just not interesting. At least it shows there is this wide gap here. You can look at this and see that we had this big accumulation of names in the 19th century and then things kind of leveled off. You can see a little bit of the history there. What our data set allows is, so for that same species, Coelochurum is Africa, I have this history of, here are these publications in these years, is what those are there, where it was treated as a real thing, like a valid name out in the world, and here's ones where it was synonymized. So I can actually, let's just superimpose that on the whole history, and let's just fill in the years in between. So here's the years where it was in use, here's the years where it's not, go all the way back. It wasn't in use before it was described. Y'all follow what I'm saying there? And I can do that with every species in there, and now in every year I just can take the sum across here and find out how names were actually in use, not just project the past or the present back to the past. This is what it looks like. And I can see on here all kinds of interesting history here. I won't bore you with it because interesting is probably pretty subjective. What I can see when I look at this, these are 4 publications, these 2 by Simpson and Haas, and this one by me and Kevin here, where publications were dealing with all species on Earth. That's why you have these crashes here, because we can sort of zero out any names that maybe they didn't mention. We'll just assume they were intending to get them all, so they must just assume those aren't useful names. So we can sort of zero those things out, so we can see the history here. You can see how people just keep using names that don't mean anything all the time. This is a human endeavor, so that's why it's messy. One of the really interesting things I found with this data, I did it just like I described to you, except if a name hadn't been used in 20 years, I didn't just keep propagating it forward, no one talked about it, it dropped off the list. And what you actually find when you explore the data this way and how we came to these species, in Haas's work right here in 1969, this is a big one, and there's actually this gap where we drop down and the majority of species were not talked about for decades. They disappeared from the literature until Haas brought them up again, and then even with that 20-year cutoff, they disappear again afterwards. Our data give us a good answer for that. If we look at just the history of publications here in blue per decade, and the history of taxonomic opinions, just where we're getting all these different taxonomic opinions, yeah, World War I and World War II, that's why no one was talking about them. They had other things to do while that was going on, and we have this huge lull. You can also see we have this incredible, just exponential takeoff in publications and taxa being talked about. We can blame the internet for that, but it's also, you know what, the Endangered Species Act. Now people care about these things. We have the easy availability of data, so that this really gives us some information about how these things are, how these names came to be. And it gives us insight into why they're named the way they're named. I'm going to skip over this because I don't want to run out of time, because I want to show the fun part here. So, to learn about Africa right here, if we want to learn how we can find out how species are distributed and things like that, we could go to Africa. That's super fun. I've done that. If we had time, I could tell you about how I was picked up by the police on that day, but I don't have time. But I've also been to all of these collections, and basically we have just photographed all the specimens there, digitally photographed them. Okay? And captured what they look like, where they were found, all the original labels on these things. We have about 30,000 specimen records, and we've assembled those into, again, a database that is connected with the database of taxonomy, so we can keep track of how these things have been named, how people have used those names. We can take the locality here in Kisangani, which is the new name for Stanleyville, and normalize those things. We can georeference that sort of information. So we can do a lot more with these records. For example, we can use the Google Static Map API, and we can make a map for all of our taxa here. We also make all of our images available to whoever wants to use them so that people can work with these specimens. FileMaker, and the reason we like it is one of the things that it's done for us here is that it allows us to automate a lot of tasks. So one of these things, I just have it running right here, is what we call an image pile. So basically, I can take any found set that we have here. This is all this records from Kisangani. There's only 446, not that many. And it basically just opens up every image, every specimen in its own little window that we can play with on the computer. So in the olden days, every museum just has a giant table and you could bring these shells out and play with them. Now we can actually just do it with these. So instead of being in one collection dealing with this, this is all of the collections, all of the specimens, and knowing what is available, what to work with, being able to separate these things out into different species and see those patterns in real time. If we look at Africa, there's really not that much data. If we georeference all the specimens that are available, it's only 5,600 different museum lots all around the world. And I've just mapped these on here, sort of the size of the dot is how many records we're talking about. So lots of these are just like one mussel was found there. Some of these are a lot more common. So we can look at the patterns there and see how sampling has been very clumped. You might think, well, that's just because there's not mussels here. No, it's just because in certain places it's easier to get to. And that's why people have sampled there. So there's a lot more work to do there. I'm going to skip over that so we don't run out of time and get to the point. So number one, we've made this infrastructure to work with what we're working with, and it's very easy to just then expand that out into other areas. So we're doing the same thing for South America. This is a great project for students. So students say they want to do something, and we just give them a part of the world. Here's Central America. We have all that stuff. We haven't looked at it yet. Just to put Africa into context, Mary and Ryan and Carissa worked on Minnesota, Wisconsin, and the Upper Peninsula of Michigan. Remember, Africa had 5,600 records. For Africa, this gigantic continent, we have 45,000 records for just this area. It's much more well sampled, so it's also a data problem. What I'm very proud of here is the fact that other researchers, not us, are using our website. This is their work, but they're just using our data. So we've put that online so that the MUSSEL Project Database has evolved from just an analysis tool that we made that's now on the web, and other people are using it as a resource. So it's the money the National Science Foundation gave us $500,000, but it's paying for other people to get work done. So, take-home message here, if you know your organisms and you can make big data sets and you can code, then there are still lots and lots of questions to answer, but you also have to not be afraid of crocodiles and hippos and schistosomiasis and stuff like that. So I think I have like a minute or two for questions. I'm happy to take any if there are any. Yes?
When you showed that map of Africa, I noticed there was like nothing in Madagascar, like 2 mussels
Yes.
in Madagascar. Why don't you go there?
There aren't any mussels there.
Really?
Yep.
How do you know?
Because I can tell you, so we have sampled in collections. There are literally 7 lots of specimens that are present there. So 7 in different collections. They're all actually from one collecting event that was then sold specimens all over the world. And they're all of a species that is just widespread throughout here. So it's probably just introduced one population. This seems like a short distance, and Africa and Madagascar just seem to go together naturally, but actually in terms of continental drift and those sorts of things, these have been, Africa and South America have only been separated for like 100 million years. This is for twice that long. So this is a giant gap here for freshwater organisms.
Thank you.
Mm-hmm. Yes?
Is that all, everything we show for resources, is that all open data people have access to?
Yes, yes. We don't, we just give sort of the specimen records right now so that it will have the verbatim locality there, and then it'll map for that. What we usually do is when people want data, so they'll say, oh, we need this for Brazil, they ask us, and then we give that. Yes?
Have you thought about using like computer vision to recognize the mussels?
Yes. The problem is, so you can imagine here we're dealing with a thing that has no countable parts, right? It's just sort of an oval. They're very amenable to photography. That's why this works. Mussels and plants, because they lay flat, they don't roll when you put them down. There's so much variation, right? And we can do very sophisticated analyses where we're not just trying to do automated from picture. You know, to get 30,000 pictures, you go through them quickly and they're not very good. You make very nice pictures. There's so much variation within and among species that it tends not to discriminate very well. If you know where you are and what species are present, then algorithms work very well for recognizing those things. Okay, thank you.